The northern end of Spencer Gulf in South Australia is not, on first sight, the kind of place one would expect to find a major natural history event. The coastline near the town of Whyalla is industrial, a working steelworks, a shipping jetty, a hydrocarbon processing plant at Port Bonython, whose flare stack is visible for miles. The water along this stretch of coast is cold in winter, dropping into the low teens Celsius by June, and the bottom is broken rocky reef, fractured into ledges and shallow caves that catch the surge from passing currents. There is no postcard quality to the place. The landscape is the practical, slightly weathered landscape of a regional Australian industrial town. And yet, every winter, between late May and early August, the few kilometres of shoreline between the Point Lowly Lighthouse and the small headland at Black Point host one of the most concentrated reproductive events in marine biology. The animals are giant Australian cuttlefish, Sepia apama, and they come to this particular reef in numbers that have no parallel anywhere else in the world, for the single purpose of mating once and then dying. The biology that drives them there is not fully understood. Sepia apama is found along the entire southern coast of Australia, from Western Australia across to New South Wales, in solitary or small groups. Most of its range, most of the year, is unspectacular cuttlefish habitat. Individuals living alone, hunting, hiding, growing. But in winter, the population that lives in the upper Spencer Gulf converges. The fractured rocky reef at Point Lowly is suitable for what cuttlefish mothers need most, which is hard substrate with sufficient crevices to receive eggs and protect them from predators. Other cuttlefish populations elsewhere along the coast presumably find smaller equivalents nearby and breed in dispersed locations. The Whyalla population does not. They travel, gather, and concentrate. And what happens during the 8 weeks they are gathered is unlike anything that happens to any other species of cuttlefish anywhere else. At the peak, recorded most precisely during a 1999 survey, the aggregation contained roughly 180,000 animals along about 60 hectares of reef. To put that density in some kind of perspective, 60 hectares is about the size of 60 rugby fields, which means roughly 3,000 cuttlefish per field. Divers descending into the water reported the sensation of being surrounded, cuttlefish on every rock, in every crevice, hovering at every depth from the surface down to the limit of available reef. The display behavior was constant. The animals were not still. They were communicating, posturing, courting, fighting, mating, and laying eggs, all at the same time, all in the same shallow water, in numbers that have not been documented before or since at this scale of cephalopod aggregation. The first thing to notice about the assembled population is its sex ratio. Among breeding Sepia apama at Whyalla, males outnumber females roughly 4 to 1. On certain days and at certain spots, the ratio is much higher. 11 to 1 has been recorded. This imbalance is the engine that drives everything else about the aggregation. With so many males competing for so few females, the contest for reproductive access is intense, and the strategies that have evolved to win it are unusually elaborate. The simplest strategy belongs to the largest males. A mature male giant Australian cuttlefish can reach nearly a metre in mantle length and 10 kilograms in weight, the size of a small dog, as has been mentioned, and substantially larger than any female he is competing for. The largest males adopt the conventional approach. They locate a female who has not yet completed her mating, position themselves beside her, and guard her against rival males through a combination of physical posture and visual display. The display is striking. The guarding male produces a bold zebra-like striping pattern across his arms and mantle, alternating dark and light bands, sharp-edged, high contrast. He directs this display at any other male who approaches. The zebra pattern, in the local cuttlefish dialect, says approximately what a snarl says in a wolf pack. Come closer if you wish, but be aware that I am willing to escalate. Most rivals, on receiving this signal, do not escalate. The guarding male holds his position. The female accepts or declines his subsequent mating attempts according to her own criteria, which, as researchers documenting the aggregation have observed, lean toward rejection in roughly 70% of cases, regardless of which male is making the approach. Reproductive success at Whyalla, even for the largest and most aggressive males, is not assured. This would be a fairly familiar story, a story about size and dominance and contested access, if it were the whole story. It is not. In 2005, a paper published in the journal Nature, authored by Roger Hanlon and a team including Marie-Jeanne Naud, Paul Shaw and Jonathan Havenhand, documented something that changed how cuttlefish reproduction was understood. The team had spent multiple seasons diving at Whyalla, filming mating sequences, identifying individual animals, and, critically, collecting DNA samples from eggs laid at the end of each successful mating. They could, by genetic analysis, determine which male had actually fertilized any given clutch. And what they found when they correlated the DNA evidence with the filmed behavior was that the smallest males in the aggregation were succeeding far more often than the visible dominance hierarchy would have predicted. The smallest males were doing this by impersonating females. The mechanism, when filmed at the appropriate speed, is one of the more remarkable behaviors documented in cephalopod biology. A small male, call him a sneaker in the technical language, approaches the area where a guarding male is occupied with his zebra display, repelling a rival. The sneaker, well aware that he will not win any direct contest with the guard, performs a transformation. He drops the male coloration entirely. He arranges his arms in the relaxed posture characteristic of an unattended female. He produces on his own skin the pattern that female sepia apama characteristically display at the aggregation, a mottled, less aggressive arrangement of color and shape. The transformation takes roughly 2 seconds, in that condition visually indistinguishable on the available evidence from an actual female. The sneaker swims unmolested past the guarding male. The guard does not register him as a threat. The guard's attention is on the rival he is already displaying at, and the female-coded animal moving across his peripheral vision is not, by the visual cues he is using, a competitor. The sneaker reaches the actual female. At close range, he drops the female disguise, sometimes in the space of a single second, and presents himself suddenly and recognizably as a male. The female has the option, as she does with any male, to accept or reject the approach. The genetic analysis from the 2005 paper showed that the sneaker's approaches succeeded in roughly 60% of attempts, a strikingly high rate when set against the 70% baseline rejection rate. The females, in other words, were not merely tolerating the deception. They were, on the available evidence, selecting the deceptive males at higher rates than the conventionally aggressive ones. The trick was not only fooling the guards, it was, in some way, working on the females. The behavior raises a question. How can a cuttlefish change skin patterns so rapidly and with such precision that another cuttlefish, an animal with the same visual system, watching from a meter away, cannot distinguish it from a member of the opposite sex? The answer lies in the chromatophore system already described, and in particular in the directness of its neural control. There is no slow change involved in the transformation. The sneaker is not waiting for hormones to take effect or for skin tissue to remodel itself. He is firing a different pattern of motor neurons. The skin responds immediately. The sex change, behaviorally speaking, is a software switch rather than a hardware modification. The same animal in the space of 2 seconds can present as either gender and switch back. Researchers documenting the behavior have recorded individual sneakers shifting back and forth as many as 10 times in 5 minutes, depending on the social conditions around them and the targets they are tracking. It is worth pausing briefly on what this means for the standard frameworks that have been used historically to describe the behavior. Popular accounts in the years after the 2005 paper tended to reach for the language of cross-dressing, for terms like transvestite male or cuttlefish drag queens. These framings, while colourful, miss the biological point. The sneakers are not expressing a stable alternative identity. They are deploying a temporary tactical signal, switchable on demand, in service of a single goal, gaining mating access in a population where direct competition is unwinnable. The behavior is, in technical terms, an alternative reproductive tactic, and it appears in similar form across many species. Small bluegill sunfish do something analogous, certain garter snakes do something analogous, various crustaceans and insects do something analogous. What is distinctive about the cuttlefish version is the speed. No other animal in the documented literature can switch sexual coloration this fast, this precisely, and this reversibly. The cuttlefish does it because the cuttlefish has the skin to do it. The behavior is enabled by the body. The Whyalla aggregation, in its peak years, was producing this kind of behavior continuously, across hundreds of mating interactions per day in the small section of reef divers could survey. The visual texture of the aggregation in the films from the late 1990s and early 2000s is overwhelming. Zebra-striped guards on every rock, sneakers materializing and dematerializing through the gaps, females, the apparent prize, moving deliberately through the crowd and making selections that bear only partial relationship to any of the displays directed at them. Eggs accumulating in the crevices below, pale, gelatinous capsules, each containing a single developing cuttlefish, each darkened by the mother's own ink to camouflage them against the rock. The aggregation, taken as a whole, is a working portrait of how Sepia apama manages the problem of reproducing once and for all in the 8 weeks available to it before death. And then, in the years after 2009, the numbers began to drop. Not in the species range as a whole, but at this particular site. In 2012, a survey of the same 60 hectares of reef returned an estimate of around 18,000 animals. By 2013, the count had fallen below 14,000, a reduction of more than 90% from the 1999 peak in just over a decade. The decline was abrupt enough to alarm marine biologists, abrupt enough to prompt a fishing ban in 2013 and a sustained investigation into what was happening. The investigation produced no single answer. The combination of factors, commercial harvest pressure through the late 1990s, possible contamination from the Port Bonithon industrial facility, natural population cycles in a species with a 1 to 2 year generation time, is still being unpicked. The aggregation has partly recovered since the fishing ban, but has not returned to anything near its peak numbers. What is happening at Whyalla year after year is now both smaller in scale and less certain in its future than it was a quarter century ago. The biology of Sepia apama has not changed in any meaningful way in the last 25 years. The female mimicry is still happening. The zebra displays are still happening. The sneakers are still slipping through the gaps. What has changed is the number of animals available to do any of this, and that change, if it continues, will end the only known event of its kind in the world. The cuttlefish, in their individual lives, have no awareness of any of this. They show up at the reef because their bodies are programmed to. They do what they do for the 8 weeks they have. And then they die, and the eggs they have left behind, glued to the rock and darkened with their own ink, develop in the cold water through the spring and hatch the following summer. The death itself is not abstract. The death is what gives the rest of the behavior its meaning. In the Lembeh Strait, off the northern coast of Sulawesi in eastern Indonesia, the seafloor is mostly volcanic sand and silt, a soft, dark surface mineralized to the color of wet ash, scattered with debris from the volcanic islands that surround it. This is not a coral reef environment in the conventional sense. There is little hard structure. The visibility, in many seasons, is poor. Divers who come to Lembeh come for what is called muck diving, looking slowly across an apparently featureless grey bottom for the small specialized animals that have evolved to inhabit exactly this kind of soft substrate. Octopuses with bizarre body plans, nudibranchs in colors that should not exist underwater, frogfish that resemble pieces of broken rock, and sometimes in the late afternoon when the light filtering through the silty water has gone amber, a small animal that walks across the bottom on the tips of its arms, displaying a series of colors that, in the absence of any obvious threat, function less like camouflage and more like a public announcement. This is the flamboyant cuttlefish. Its scientific name, until very recently, was Metasepia pfefferi. In the taxonomic revision of 2023, the genus was reorganized, and the species is now formally Ascarosepion pfefferi, though the older name remains in nearly every popular reference, on aquarium signage, and in the active vocabulary of working researchers. By any name, the animal is unusual. It is small. Adults reach perhaps 8 or 9 centimeters in mantle length, smaller than a human hand, and the most immediately striking thing about it is its coloration. Where most cuttlefish hold themselves to a measured palette of browns and greys and mottled neutrals, working hard to disappear, the flamboyant cuttlefish does the opposite. It pulses through a vocabulary of bright yellows, deep purples, reds saturated almost to the limits of what its chromatophore system can produce, and brilliant whites, sometimes all of these at once, in shifting bands across its body, often paired with the upraised display of its lobate arm membranes. The behavior is so unlike standard cuttlefish behavior that, for many years, it was treated as a curiosity rather than a clue. Until, in the early 2000s, the Australian cephalopod biologist Mark Norman, working out of Museum Victoria in Melbourne, ran tissue samples through a toxicology assay and discovered that the muscle tissue of the flamboyant cuttlefish appeared to be toxic enough on his initial measurements to be lethal to a human at sufficient doses. This finding, picked up in the science press and in the natural history documentary Kings of Camouflage, transformed the popular understanding of the species. The flamboyant cuttlefish was now described in many sources as the world's only known venomous cuttlefish, comparable in toxicity to the blue-ringed octopus, an animal that has killed people. The colorful displays on this account were no longer puzzling. They were warning signals, the marine equivalent of the bright yellow and black stripes of a wasp, advertising to potential predators that this small, slow-moving animal was not worth eating. The story was satisfying. It made evolutionary sense. It explained in a single move why an animal with such an obviously inadequate cuttlebone, too small to support buoyancy, too small even to give the animal an effective swimming gait, leaving it walking across the bottom because it has no real alternative, could afford to be so brightly colored without being eaten. Toxicity bought it the freedom from camouflage. The bright colors were the receipt. Subsequent work has complicated the picture. A follow-up study by researchers at Georgia Southern University and Utah State University Uintah Basin, looking for the specific neurotoxin, tetrodotoxin, the molecule responsible for blue-ringed octopus toxicity, found it in the flamboyant cuttlefish at only trace concentrations. Concentrations, the researchers concluded, far too low to be toxic to any vertebrate, including a human. The early reports of dangerous toxicity may have been overstated or may have been based on a different toxic compound that has not yet been characterized. The current consensus, as of 2026, is that the flamboyant cuttlefish is mildly toxic at most, uncomfortable to eat perhaps, but not lethal, and that the relationship between its bright coloration and its chemical defense, if there is a stable relationship, has not yet been adequately mapped. The colors remain, the walking remains, the cuttlebone too small to swim with remains. The explanation, the part everyone thought had been settled, is open again. The flamboyant is one species. The cuttlefish family contains roughly 120, and each of them carries its own peculiarities. Some are worth noting briefly, not as a survey, but as a reminder that the things this script has been describing, the camouflage, the strike, the cognition, the polarized signaling, are the family's shared inheritance, expressed in different bodies under different selective pressures, generating different specific solutions. The morning cuttlefish, Sapia plangon, is one of these. It lives along the eastern coast of Australia, in shallow coastal waters, in a population that has been studied carefully over the last 15 years by researchers including Cullen Brown at Macquarie University. The morning cuttlefish does something that, in 2012, when it was first carefully documented, surprised the field. During courtship, when a male is approaching a female while a rival male watches from the other side, the morning cuttlefish produces two patterns on its body at the same time, one on the side facing the female, one on the side facing the rival. The side facing the female displays the standard male courtship pattern. The side facing the rival displays a pattern characteristic of an uninterested female. The body, in other words, is partitioned. The two halves are running different communications to different audiences simultaneously, and the animal, by holding its orientation precisely, can sustain the dual signal across an entire courtship encounter. The chromatophore system is asymmetric. The neural control is asymmetric. The morning cuttlefish is, in effect, telling two different stories at once with the same skin. Then there is the dwarf cuttlefish, formerly Sepia bandensis, and now, post-taxonomy, Ascarosepion bandense. The dwarf cuttlefish is small, native to the Indo-Pacific, and in the last several years has become the cephalopod of choice for laboratory neuroscience. The reasons are practical. Most cuttlefish are difficult to keep in captivity through multiple generations. They are slow to reach maturity, demanding in their requirements, hard to breed reliably. The dwarf cuttlefish is faster. It reaches sexual maturity in about 4 months. It tolerates laboratory conditions better than most of its relatives. A research lab at Columbia University, led by the neuroscientist Tessa Montague, has built an entire research program around the species, culturing successive generations, mapping the dwarf cuttlefish's brain in 3 dimensions, building an interactive online atlas of its neuroanatomy, freely accessible to other researchers. The dwarf cuttlefish in 2026 is well on its way to becoming for cephalopod neuroscience what the zebrafish is for vertebrate neuroscience, the model organism on which the field's foundational questions get worked out, which means that over the coming decade, more is likely to be learned about the neural architecture of a cuttlefish brain than has been learned in the entire previous history of the field. The cuttlefish family, taken as a whole, is a study in evolutionary variation around a single body plan. The plan is conservative: 8 arms, 2 tentacles, the W-shaped pupil, the chromatophore skin, the chambered cuttlebone. These elements appear, with minor adjustments, in every species. What varies is the way each species has used the plan to occupy a particular environment. The giant Australian cuttlefish has scaled up to the size of a small dog and adopted seasonal aggregation. The flamboyant has shrunk, abandoned swimming, and possibly evolved chemical defense. The mourning cuttlefish has learned to talk in two directions at once. The dwarf has been miniaturized and accelerated generation after generation until it has become the field's most tractable laboratory subject. Each species is its own solution to a slightly different version of the same problem. The problem, in every case, is how to live as a small, soft-bodied predator in shallow coastal water, surrounded by predators of one's own. And every species, despite the variation, ends in the same way. 1 year, maybe 2, a single reproductive event. The female lays her eggs, attaches them to whatever substrate is appropriate to her habitat, darkens them with her own ink, and stays with them as long as her body permits. The body, by the time the eggs are attached, has already begun to deteriorate. The hormonal cascade of reproduction triggers, in cuttlefish as in salmon and many other semilparous species, an irreversible process of decline. The animal will not feed again, or will feed only minimally. Its muscle tissue will break down to provide energy for the final tasks. Its chromatophores will begin to fail. Within weeks of egg-laying, the female cuttlefish, whether dwarf or mourning or giant Australian or flamboyant or common European, will be dead. The eggs will hatch some weeks later. The hatchlings will emerge already pigmented, already camouflaging, already equipped with the inherited neural architecture that, with time and experience, will let them do what their mothers did. They will not know their mothers existed. The transmission across cuttlefish generations is purely genetic. Whatever the mother had learned, whatever specific past events her body had been holding, ends with her. This pattern is, in evolutionary terms, an unusual choice. Most animals that live long enough to learn anything important transmit at least some of what they have learned to their offspring, through teaching or social observation or extended care. Cuttlefish do not. Each generation starts over from scratch, equipped with the species' shared inheritance and nothing more. Each individual cuttlefish learns within its short window, becomes whatever it becomes, and then takes the contents of that learning with it when it dies. The pattern repeats, has repeated for tens of millions of years, and will continue to repeat as long as cuttlefish exist. And for tens of millions of years, the pattern has included one of the more curious features of cephalopod biology, a behavior the animal performs every day, often for hours at a time, that may or may not be a form of dreaming. In 2019, a paper appeared in the Journal of Experimental Biology that, if its conclusions hold up to the additional scrutiny they are now receiving, will require a careful adjustment to how biology has understood one of the most universal behaviors animals do. The paper was authored by Teresa Iglesias and her collaborators, including Jean Boal at Millersville University, the neuroscientist Marcos Frank at Washington State University, the vision researcher Jochen Zeil at the Australian National University, and Roger Hanlon at Woods Hole. The subject was Sleep in Cuttlefish. The title, Cyclic Nature of the REM Sleep-like State in the Cuttlefish Sepia officinalis, gives away the finding, but does not quite convey what it feels like to read the paper for the first time, or to watch the video footage it draws on, which the authors made available alongside their published data. The footage was taken at night in a laboratory in Pennsylvania of a single common cuttlefish sitting at the bottom of its tank. For most of the recording, the cuttlefish does nothing. It rests on the substrate, its body color faded to a uniform pale tone, its pupils closed to thin slits. Sometimes the fins along its mantle drift slightly with the water circulation. Otherwise, the animal might as well be a stone. This is the state the researchers came to call quiet sleep. By every behavioral criterion that biologists use to identify sleep across the animal kingdom, the cuttlefish in this state was sleeping. Its arousal threshold was elevated, It took stronger stimuli to rouse it than it would take in waking. Its movements had ceased. Its body had assumed a posture characteristic of the state and not of waking activity. And, critically, if the animal was prevented from entering this state for a prolonged period, it compensated afterward with extra time in the state, a phenomenon called rebound sleep that biologists treat as one of the most reliable signatures that an animal is in fact sleeping rather than merely resting. The cuttlefish, in other words, sleeps. This was already known. The 2019 paper was not breaking new ground on the basic question. What the paper was documenting with new precision was something the field had noticed in passing but had not characterized carefully. A second state occurring at regular intervals throughout the cuttlefish's sleep period, in which the animal's body behaved very differently than during quiet sleep. The transition was abrupt. After approximately 30 minutes of quiet sleep, the cuttlefish on the recording and the other animals the researchers monitored, 11 in total over the course of the study, would shift within seconds into a state the team named active sleep. The body did not get up. It did not begin moving in any purposeful way. But the chromatophores along the skin began to activate, not all at once and not in the coordinated patterns of waking camouflage. The activations were sporadic, asymmetric, sometimes appearing in small bursts across one section of the body, sometimes traveling across the mantle in slow waves. The arms, which during quiet sleep had been still, began to twitch. Brief contractions, small movements that did not propel the animal anywhere. The eyes, which during quiet sleep had been closed, began to move rapidly behind the closed pupils. The whole sequence lasted, on average, 2 minutes and 25 seconds, and then, as abruptly as it had started, it stopped. The cuttlefish returned to quiet sleep. 30 minutes or so later, it happened again. The pattern repeated through the night, on a cycle of roughly 34 minutes of quiet sleep, followed by 2.5 minutes of active sleep. The cuttlefish spent, in total, about 30% of every 24-hour period in some form of sleep, roughly the same proportion of time, as it happens, that an adult human spends asleep on a typical schedule. Of that sleep, approximately 7% was in the active state. The rest was quiet. The team did not, in the 2019 paper, claim that the cuttlefish was dreaming. They were careful not to. The word does not appear in the paper's main text. What they claimed was that the cuttlefish was showing behavioral signatures, the rapid eye movements, the muscle twitches, the cyclic alternation with a longer quiescent state, that are the standard markers used to identify REM sleep in vertebrates. In vertebrates, those markers reliably indicate the state of sleep during which dreaming, when it occurs, takes place. Whether the same markers appearing in a mollusk indicated the same underlying neural activity was a question the paper raised but explicitly left open. The implication, though, is hard to ignore. REM sleep, the state of rapid eye movement, the state in which the body's muscles are partly paralyzed while the brain runs through what is, on the dominant scientific account, some form of offline neural activity related to memory consolidation or to other not yet understood cognitive functions, was thought for most of the 20th century to be a vertebrate phenomenon. It had been documented carefully in mammals, in most birds, and in the 2000s in certain reptiles. It had never been documented in an invertebrate. The accepted explanation, until recently, was that REM sleep had evolved once, somewhere in the early history of warm-blooded animals, as a feature tied to the metabolic and neural complexities of the vertebrate brain. The cuttlefish would seem to complicate that account. The cuttlefish has no vertebrate brain. It diverged from the lineage producing vertebrates more than 500 million years ago into a separate evolutionary trajectory that has produced none of the structures REM sleep is supposed to depend on. If the cuttlefish is doing something neurally equivalent to REM sleep, then either REM sleep evolved twice, once in vertebrates, once in cephalopods, or the trait is much older than the lineage that contains its better-known examples, dating back to a common ancestor so distant that essentially every animal on Earth might share some version of it. Subsequent work, including a 2023 Nature paper on a similar active sleep state in octopus, has begun to push the field toward the more interesting of these two possibilities. The octopus paper, in particular, did go further than the Iglesias team had been willing to go, recording neural activity in addition to behavior and finding that the brain during active sleep showed patterns of electrical activity that, in a vertebrate, would be straightforwardly classified as wake-like. The octopus, during its equivalent of REM sleep, has brain activity that resembles being awake, except the body is not awake. The octopus is somewhere else. Where exactly is not known. The question of whether a cephalopod can experience anything during active sleep, whether the animal in some sense sees images or feels sensations or revisits past events, is one of those questions that the existing scientific methods are not yet built to answer. The behavioral evidence is suggestive. The neural evidence, in the octopus case, is more suggestive still. The cuttlefish footage in particular has a quality that has affected many of the researchers who have watched it carefully. The chromatophores on the sleeping animal cycle through patterns that, in a waking cuttlefish, would be specific signals, hunting displays, courtship displays, agonistic displays. The animal in active sleep does not perform any of these patterns cleanly. Instead, fragments of them surface briefly in the wrong sequences, a hunting flash appearing, then dissolving, a courtship coloration partly visible, then replaced by something unrelated. The skin, as one of the paper's authors described it in a later interview, looks like an animal cycling through its own behavioral vocabulary at random, the way a sleeping human might mumble fragments of an unconnected conversation. Whether this is what is happening, whether the cuttlefish, in some unmappable sense, is replaying its day across its skin, is a question without an experimental method capable of answering it. What can be said is that the cuttlefish, every night of its short life, spends roughly 30 minutes at a time in a state during which something is going on inside it that we cannot see, punctuated every half hour or so by 2-minute windows in which fragments of that something become visible on its body. Every cuttlefish in the world, in every ocean, every night is doing this. The behavior is universal across the species that have been carefully studied. It is presumably ancient. It is happening in millions of individual cuttlefish at this moment, in every cuttlefish that is currently between meals and not actively threatened. They are sleeping in the way the species sleeps, and within that sleep, they are doing something else that the species also does, and whatever that something is, it leaves visible traces on the skin every 30 minutes for as long as the animal lives. The first cuttlefish that did this, the first ancestor of the modern cuttlefish that had a sleep cycle of this shape, with these two phases on these intervals, lived in the ocean of the late Cretaceous, in the world that existed shortly before an asteroid arrived off the coast of what is now Mexico and rearranged the surface of the planet. That cuttlefish would have been sleeping when the asteroid hit. So would the second one and the third one. And every cuttlefish since then, through the 66 million years that followed, has been doing approximately the same thing every night. This is not the oldest behavior in the cuttlefish's repertoire. The oldest, in fact, predates the family entirely. And to find its origin, one has to travel further back than the asteroid, further back than the dinosaurs, into a world that the cuttlefish's ancestors inhabited when most of the animals alive today did not yet exist in any recognizable form. In the limestone quarries near Maastricht, In the southern Netherlands, the rock weathers a particular shade of pale yellow under exposure to air. The stone is soft enough to be cut with a saw and dense enough to have been used for centuries as a building material. The older quarters of the city are built from it. The cathedrals of the surrounding region are faced with it, and the underground galleries left behind by the cutting operations now extend for tens of kilometers beneath the local farmland. The rock is also, as geologists have known since the early 19th century, one of the more important fossil deposits in Europe. It records the conditions of a shallow tropical sea that covered this part of the world in the last few million years of the Cretaceous period, in the time just before the asteroid impact that ended the age of the large dinosaurs. The rock takes its name from the city. The geological stage it represents, the Maastrichtian, takes its name from the rock. And among the many fossils that have come out of these quarries over the past 200 years, in 1965, a paleontologist named Doris Meyer described one that had been overlooked for decades. A small, ovoid, chambered structure, weathered but intact, that turned out, on careful examination, to be the internal shell of a cuttlefish. The fossil was given the name Ceratisepia elongata. It is approximately 68 million years old. It is, as of the time of writing, the oldest unambiguous cuttlefish fossil ever recovered. The internal architecture, the chambered structure, the connecting subuncular zone, the geometry of the supporting pillars is recognizable. Set beside the cuttlebone of a modern Sepia officinalis, it does not look like a primitive prototype that would later evolve into the structure we know. It looks like the structure we know. The pillar network is there. The chamber spacing is there. The dorsal shield is there. 68 million years separate the fossil from the cuttlebone in a pet shop today, and the engineering between them has, in any visible sense, not been improved on. This is the kind of fact that, on first encounter, does not seem like it can be right. Most animal lineages looked at across 68 million years have changed substantially. The mammals alive then were small, generalized, mostly nocturnal. Nothing like the elephants and whales and humans that have descended from them. The birds alive then were not yet recognizable as members of the modern families. The plants alive then were different in composition and distribution. The world itself was different. The continents were in slightly different positions. The climate was warmer. The shallow tropical sea that covered Maastricht has long since drained, the land has risen, and the quarries that now expose the fossil sit 200 kilometers from the nearest ocean. Everything has changed, and the cuttlefish, by every available measure, has not. The fossil record of cuttlefish is incomplete. Cephalopod soft tissue almost never preserves, and the cuttlebone, though it does preserve being mineralized, is delicate and easily destroyed. Most of what is known about the deep history of the family comes from molecular clock estimates rather than from direct fossil sequences. The molecular methods compare the genetic divergence between living cuttlefish species, calibrated against known fossil dates from related cephalopod groups, and produce estimates of when various branching events must have occurred. The estimates have been refined over the last 15 years as the techniques have improved, and they now converge with reasonable confidence on a single picture. The cuttlefish lineage, as a distinct evolutionary branch separate from the squids and the octopuses, appears to have originated somewhere around 88 million years ago during the second half of the Cretaceous period. The split from the squid lineage was earlier. The two groups had been on independent trajectories for tens of millions of years before the cuttlefish form took its modern shape. The split from the octopuses was earlier still. By the time of the asteroid impact, 66 million years ago, cuttlefish in something close to their modern form were already swimming in the world's shallow seas. They survived the impact. The dinosaurs did not, the ammonites did not, 3/4 of marine species did not, and the cuttlefish, small, soft-bodied, dependent on coastal habitats, did. Why is not fully understood. Their reproductive cycle is short, which would have helped any species rebuild quickly from a survivor population. Their habitat range is broad, which would have spread the risk across many local ecosystems. Their cognitive flexibility is high, which would have helped them adapt to whatever post-impact conditions emerged. Some combination of these factors probably carried the lineage through the worst extinction event of the last 500 million years. Whatever the mechanism, the cuttlefish came out the other side. In the years since 2020, a new technique has begun to expand the cuttlefish fossil record in unexpected ways. A group of paleontologists working with machine learning specialists has developed a procedure they call digital fossil mining. Using AI image recognition to scan through enormous archives of digitized fossil photographs and museum specimens, looking for objects that may have been misidentified or overlooked at the time of their original collection. In 2025, a paper published using this method described a new specimen recovered from the Upper Cretaceous of South Dakota, a small fossil, approximately 73 million years old, that the team named Uluciala rotundata. The animal turned out to have a body plan intermediate between the modern sepioids, cuttlefish, and their close cousins, the sepiolids, bobtail squid. Its discovery has helped refine the dating of the cuttlefish bobtail split and has confirmed that the diversification of the modern sepioid group was already well underway in the late Cretaceous, earlier than some of the molecular estimates had suggested. The deeper history is harder to access. Cephalopods as a class are very old. They first appear in the fossil record during the Cambrian period, more than 500 million years ago, in forms that bore little resemblance to anything alive today. The earliest cephalopods had external shells, chambered, conical structures from which the soft body extended downward, and they were among the dominant predators of the early oceans, in a world that had not yet produced bony fish, that contained no vertebrates of any kind on land, and that consisted almost entirely of marine ecosystems organized around invertebrate body plans. For roughly the first 300 million years of cephalopod history, all cephalopods had external shells. The nautilus, a living relative of the cuttlefish, is the last surviving representative of that ancient body plan. The transition from external shell to internal shell happened sometime around the late Paleozoic era, perhaps 270 million years ago. The shell did not vanish, it was absorbed. It moved from the outside of the body to the inside, becoming a structural element rather than a defensive enclosure. This is the move that produced the coleoid cephalopods, the group that contains squids, octopuses and cuttlefish, and it appears to have happened only once. Every soft-bodied cephalopod alive today is descended from that single evolutionary event. The cuttlebone in a modern cuttlefish is the direct lineal descendant of the chambered shell of a Paleozoic cephalopod, internalized and retained, used now for buoyancy rather than for protection. The same architecture repurposed across 250 million years. What this means for the cuttlefish as a body plan is that the animal has been working on its current form for an extraordinary length of time. The chambered shell, the precise neural control of skin coloration, the camera-type eye, the elaborate reproductive behaviors, none of these arrived all at once. Each was selected for, refined, retained, and integrated across an evolutionary timescale that exceeds the entire history of mammals as a class. By the time the first true mammals appeared in the late Triassic, cuttlefish ancestors had already been refining their solutions to coastal predator biology for over 100 million years. By the time the first primates appeared, the cuttlefish form was essentially complete. By the time hominins began making tools, cuttlefish had been doing what cuttlefish do for a span of time that, expressed in human generations, is functionally infinite. There is a tendency in popular descriptions of evolution to treat older lineages as somehow simpler or more primitive than younger ones. The cuttlefish is a good reminder that this is not how evolution works. An animal that has been refining a single body plan for 100 million years is not, by any reasonable measure, simple. The skin color system is not simple. The W-shaped pupil is not simple. The chambered cuttlebone is not simple. The episodic memory, the polarized signaling, the 8 arms with their independent musculature, the central brain that coordinates all of it, none of these is simple. The cuttlefish is one of the most elaborately solved problems in the history of life on Earth. The solution was completed long ago. It has been maintained with minor adjustments ever since. The fossil from Maastricht sits in a museum drawer somewhere in the Netherlands. The cuttlefish in the aquarium sits on a piece of artificial substrate. The two animals are separated by 68 million years and by no meaningful change in body plan whatsoever. The species range has shifted, the genus assignments have been revised, the specific habits have varied. The underlying engineering has not. What is alive in shallow water tonight is what has been alive in shallow water for most of the time since the dinosaurs ended, doing approximately what it was doing then. Whether it will still be doing it in another 68 million years is a different question, and one that, on the present evidence, is harder to answer than it should be. In 1999, the breeding aggregation at Point Lowly contained roughly 180,000 cuttlefish. The number was arrived at through careful underwater transect surveys conducted across the 60-hectare site by researchers who had been studying the aggregation since the late 1990s and who, by the late 1990s, had developed reliable methods for estimating cuttlefish density across the broken rocky reef where the animals gathered. The 1999 count was the highest ever recorded at the site. It was also, as it turned out, the peak. In 2012, the same surveying methods conducted across the same 60 hectares returned a figure of approximately 18,000. In 2013, the number was below 14,000. From the 1999 peak to the 2013 trough, the aggregation had lost more than 90% of its breeding population in a span of 14 years. What caused the decline is not, in 2026, fully resolved. The most defensible answer is that several factors contributed in combination. Commercial fishing of sepia apama in the South Australian Gulf had intensified through the late 1990s, with foreign export markets driving harvest levels that by some estimates exceeded sustainable yield for several consecutive years before the regulatory response. The Port Bonython industrial facility, situated immediately adjacent to the spawning ground, had experienced 2 significant contamination events in the years preceding the decline, and the question of whether residual contaminants in the sediment may have affected egg viability remains open. Underlying both factors is the species' own biology, a 1 to 2 year generation time, a single reproductive event per individual, and a population structure that, when reduced, recovers slowly. A cohort of cuttlefish that fails to spawn cannot be replaced by holdover adults from previous years because there are no holdover adults. The animals that fail to reproduce simply die, and the aggregation depends on each new generation to refill itself. A fishing ban was implemented in 2013. The aggregation has partially recovered in the years since. Surveys through the early 2020s have returned counts in the 30 to 40,000 range, well above the 2013 floor, but still far below the 1999 peak. Whether the population can return to its earlier numbers, or whether the decline reflects a permanent shift in the local conditions, is a question that is being studied actively. The Port Bonathon facility remains operational. The genetic structure of the Whyalla population has been examined and confirmed as distinct from other Sepia apama populations along the southern Australian coast, meaning that if this particular aggregation is lost, it is unlikely to be repopulated from elsewhere. The breeding ground at Point Lowly is the only one of its kind in the world. If it ends, it does not resume somewhere else. The Whyalla situation is the most carefully documented case of cuttlefish decline currently in the literature. It is not the only one. Several cuttlefish fisheries around the world have shown declining catch trends over the last 2 decades, with the Mediterranean populations of Sepia officinalis being among the most pressured. The common cuttlefish supports substantial commercial fisheries in southern Europe. Italy, Spain, France and Portugal all maintain significant cuttlefish harvests, and the species has been one of the most heavily fished cephalopods in the Mediterranean for decades. Stock assessments conducted under the General Fisheries Commission for the Mediterranean have varied in their conclusions, but the overall picture is one of pressure rather than abundance. The species is not yet listed as threatened by the International Union for Conservation of Nature. Most cuttlefish species, in fact, are listed either as least concern or as data deficient, the latter category meaning that for many cuttlefish species, there is not yet enough population information to determine whether they are at risk. This second category is the more concerning of the two. Sepia apama itself is currently listed as near threatened, which is the conservation category one step below vulnerable. Many of the smaller and more localized species, the flamboyant cuttlefish, the mourning cuttlefish, various Indo-Pacific species, have not been assessed in detail at all. The absence of a formal threatened species designation is not, in these cases, an indication that the species are safe. It is an indication that no one has yet looked carefully enough to know whether they are not. There is a second category of threat that has been receiving increasing attention. Cephalopods are calcifying organisms. The cuttlebone, the radula, the beak, the statoliths all depend on the deposition of calcium carbonate from seawater, and the chemistry of seawater is changing. Atmospheric carbon dioxide concentrations have risen from roughly 280 parts per million at the start of the industrial era to approximately 420 parts per million in the present, and a substantial fraction of that additional carbon dioxide has been absorbed by the ocean, where it reacts with water to form carbonic acid. The result, over the last century and a half, is a gradual but measurable decrease in seawater pH, the process now generally called ocean acidification. The chemistry is well established. The biological consequences are still being mapped. For cuttlefish, the principal vulnerability appears to be developmental. Adult cuttlefish are reasonably tolerant of modest decreases in pH. Their physiology can buffer the change, at least within the ranges currently observed. The embryos and the early hatchlings are more vulnerable. Recent experimental work, including studies by groups working on Sepia officinalis in the eastern Atlantic and on Sepiella inermis in Chinese coastal waters, has shown that exposing developing cuttlefish embryos to low pH conditions delays embryonic development, reduces hatching success, and produces hatchlings with measurable abnormalities in cuttlebone calcification. The effects are most pronounced at pH levels below 7.6, which is below the current average seawater pH of approximately 8.05, but above the pH levels projected for many coastal waters by the end of the current century if current emission trajectories continue. The eye structure of juvenile cuttlefish has also been shown to be affected. Photoreceptor architecture undergoes measurable changes under sustained low pH conditions, with implications for the visual capabilities the animal depends on for nearly every behavior in its repertoire. None of this is catastrophic at present pH levels. The current concern is about the trajectory. Ocean pH has dropped by approximately 0.1 units since the start of the industrial era, which represents a roughly 30% increase in hydrogen ion concentration in seawater, and projections suggest a further decrease of 0.2 to 0.4 units by 2100 in many ocean regions. If those projections hold, cuttlefish embryos developing in late 21st century coastal waters will face conditions that, in laboratory experiments, have measurably impaired development. What this means for actual cuttlefish populations under actual ocean conditions, with the various buffering mechanisms and local variability that the laboratory does not capture, remains uncertain. The third category of pressure is habitat. Cuttlefish are coastal animals dependent on shallow waters, on hard substrate for egg attachment, and on the small invertebrates and fish that constitute their prey base. Coastal habitats around the world have been altered substantially over the last century, through dredging, through the destruction of seagrass beds and oyster reefs, through the eutrophication caused by agricultural runoff, through the spread of coastal development. The specific habitats that cuttlefish depend on for spawning, rocky substrates in particular, are not always the habitats that conservation efforts have focused on protecting. Coral reefs receive substantial conservation attention. The broken rocky reefs that, say, Sepia apama uses for egg attachment receive much less. The result is that the spawning grounds of various cuttlefish species are, in many regions, exposed to ongoing physical degradation without much specific protection. One development in the last several years has been positive. In 2021, following a comprehensive review commissioned by the United Kingdom government and led by the philosopher Jonathan Birch at the London School of Economics, working with cephalopod biologists including Alexandra Schnell, the UK Animal Welfare Sentience Act was extended to cover cephalopod mollusks and decapod crustaceans. This was the first time in any major legal jurisdiction that the legal definition of sentient animal had been formally extended beyond vertebrates. The change has practical consequences. Live cephalopods now fall under welfare provisions in UK law. Certain commercial practices have been subject to additional scrutiny. Research involving cuttlefish in UK institutions is conducted under welfare protocols that did not previously apply. The change has had ripple effects internationally. Several other jurisdictions have begun reviewing their own animal welfare frameworks in light of the UK precedent. A 2026 review paper by Schnell and colleagues updating the 2021 evidence assessment found strong evidence of sentience in cuttlefish across 6 of the 8 criteria the framework uses, a level of evidence comparable to what has been documented for many mammals. The conservation picture taken altogether is uneven. One major aggregation has declined and partly recovered. Several commercial fisheries are under sustained pressure. Many species have not been carefully assessed. Ocean chemistry is shifting in ways that will affect developing embryos. Some habitats are degrading, and the legal recognition of cephalopod sentience, while substantial in symbolic and practical terms, applies in only a few jurisdictions and does not yet meaningfully constrain global fishing practices. The cuttlefish, as a family, is not in immediate crisis. Many species are still numerous, still widely distributed, still common in their native ranges. But the trajectory of the major pressures, fishing, ocean acidification, habitat degradation, climate change, is not improving, and the animals themselves have no margin to spare. A species that reproduces once and dies cannot adapt to changing conditions across multiple generations of adult lifespans. It either solves the problem in one generation or it does not, and what does not solve it is gone. What this means, in the most concrete possible terms, is that the cuttlefish that has been the subject of this script, the species in all its diversity, in all its accumulated evolutionary refinement with all the capacities that have been described, is being asked to navigate the next several centuries through a set of conditions it has not previously encountered. It has come through difficult periods before. The asteroid was a difficult period. The various extinction events of the last 100 million years were difficult periods. The cuttlefish has, by every available measure, the cognitive flexibility and the reproductive resilience to handle a great deal. Whether it can handle what is currently being directed at it in the timescales involved is a question that the next several decades will answer. The question is being answered slowly, one breeding season at a time. At Whyalla, at this moment, the aggregation is somewhere between its 2013 floor and its 1999 peak. The animals are arriving, displaying, mating, laying their eggs, and dying. Whether their offspring will return in numbers, in 5 years, and in 50, will depend on what the surrounding human ocean decides to do. The female cuttlefish that is going to die in this chapter is a Sepia officinalis, 16 months old, weighing approximately 1.5 kilograms, residing in a small rocky cove on the southern coast of England. She is, as cuttlefish go, ordinary. She has spent her short life doing what cuttlefish do, hunting in the shallow waters around her cove, hiding from larger predators, evading the trawl nets that pass occasionally through the deeper channels offshore, and growing. She has fed mostly on small crustaceans and juvenile fish. She has, on perhaps a dozen occasions, deployed her tentacles in successful strikes on prey larger than is typical for her size class. She has been camouflaged on dark rocks, on light sand, on mottled patches of seaweed. She has slept somewhere between 5 and 7 hours of each 24, and during those hours she has cycled through the quiet sleep and active sleep states that all cuttlefish cycle through, and her skin has displayed, in the active phases, fragments of patterns whose function remains unclear. She has mated 3 weeks ago with 3 different male cuttlefish in quick succession near the entrance of the cove where she now sits. She is now attached to a rocky overhang in a shallow crevice that is about 6 meters below the surface, doing the last thing she will ever do. She is laying eggs. The eggs themselves are pale yellow gelatinous capsules, roughly the size of grapes, attached by short stalks to the underside of the rock. She has been laying them in clusters for the past 4 days, working her way along the underside of the overhang in a pattern that, viewed from above, would resemble a slowly extending grapevine. Each egg, as she releases it, passes briefly through the chamber where her ink sac empties and emerges coated. The ink darkens the capsules from pale yellow to deep brown-black. The darkening is not decorative, it is camouflage. The ink-coated eggs blend into the dark rock under the overhang, making them substantially harder for the small fish and crustaceans that prey on cephalopod eggs to detect. She has been performing this same operation on every egg for the past 4 days. She will not stop until either the eggs are exhausted or her body is. Her body is starting to fail. She has not eaten in nearly 2 weeks. Her chromatophores, normally responsive within milliseconds to any change in her surroundings, have begun to respond more slowly. Some patches of skin on her dorsal mantle have stopped responding at all. Small dead zones where the motor neurons are no longer firing, where the muscle fibers that control the chromatophore opening have begun to deteriorate from disuse and from the systemic decline that the reproductive hormones have triggered. Her muscle mass overall has dropped by roughly 40% since she began this cycle. She is breaking down her own tissues to fuel the production of eggs and the energy required to attach them, and the breakdown is not reversible. There is no point in the cuttlefish's life cycle at which a senescing female recovers and lives on to reproduce again. The reproductive event, in cuttlefish biology, is also the terminal event, the hormonal cascade that initiates egg-laying simultaneously initiates the dissolution of the body that is laying them. She does not appear from the outside to be in distress. Her arms move gently as she works. Her eyes, the W-shaped pupils now somewhat less crisp than they were a month ago, remain oriented toward whatever she is currently doing. She makes small adjustments to her position to reach the unattached corners of the overhang. She has not retreated, even when small fish pass nearby and ordinarily would have triggered a defensive response. She is committed to the rock. The eggs are everything she has left to do. Across the cove, in the deeper water beyond the overhang, the rest of her cohort is in approximately the same state. Other females, attached to other rocks, are completing their own egg deposits. Some males, having mated and now unproductive, are dying in slower form across the open sand. The cohort that arrived at this cove 8 weeks ago is dissolving, individual by individual, into the substrate. Within another 2 to 3 weeks, the entire generation will be gone. The cove will hold no live cuttlefish at all for several months. The eggs will incubate through the autumn, developing inside their darkened capsules, the embryos visible only on close inspection through the gelatinous wall. Sometime around the middle of winter, when the water has reached its coldest and the developmental clock has run its course, the embryos will hatch. 1 or 2 centimeters in body length, fully pigmented, already capable of camouflage, already capable of strike, already carrying the entire inherited repertoire that defines what it is to be a cuttlefish. They will not know that any of the adults that produced them ever existed. There will be no adults present to teach them. The transmission between the generation now dying and the generation that will hatch into the spring is genetic. Whatever this particular female has learned across her 16-month life, whatever specific memories she has accumulated of this cove, of the male cuttlefish she mated with, of the predators she evaded, of the prey she captured, will end with her. This is the pattern. It has been the pattern for tens of millions of years. Every female cuttlefish that has ever lived has reproduced once and died, and every juvenile cuttlefish that has ever hatched has begun from scratch. Take a moment, if it is useful, with what this animal is and what has been described across the previous hours. A common cuttlefish is, on any reasonable measure, one of the more remarkable products of evolutionary time. The body plan is conservative: 8 arms, 2 tentacles, a chambered cuttlebone, a brain wrapped around an esophagus, and has been working in approximately its current form since before the asteroid impact at the end of the Cretaceous. The skin contains millions of chromatophores under direct neural control, an apparatus that no other animal lineage has matched in either speed or precision. The eye has one type of photoreceptor and no capacity for color discrimination, and yet the animal produces on its skin color patterns matched to the substrate it sits on by mechanisms that science, in 2026, has not yet fully resolved. The skin itself contains light-sensitive proteins distributed across the body, opsins, suggesting that the animal may, in some still undetermined sense light through tissues that are not eyes. The retina detects polarized light, opening a channel of visual communication that humans, lacking the relevant photoreceptor geometry, do not perceive. The hunting strike takes 30 milliseconds. The brain demonstrates self-control comparable to that of a parrot or a young chimpanzee, episodic memory comparable to that of a corvid, predictive motor anticipation, that calculates the target's future position before firing. The reproductive system includes alternative tactics, female impersonation by smaller males, that work on the available evidence more often than the conventional alternatives. The animal sleeps in 2 phases, and in one of those phases, its skin displays fragments of patterns that may or may not be the externalized signature of something like dreaming. The species has survived a major extinction event. It has been refining its solutions to coastal predator biology since long before the existence of mammals as we know them. It has done all of this in a body that lives 1 to 2 years, that reproduces once, that knows neither parents nor offspring, and that ends every individual in every generation without exception at the conclusion of its single reproductive act. There is no way to summarize what the cuttlefish is that does not understate it. Most attempts to describe the animal end with the same recognition. The closer the look, the harder the categorization. We do not yet have a vocabulary adequate to the cuttlefish. The cuttlefish has not needed our vocabulary. It has been doing what it does for as long as we have been a species, and considerably longer. The female on the rock has been still for the past several minutes. Her arms have stopped moving. The eggs immediately around her are attached, dark, beginning their 2 to 3 month development. There are perhaps 24 unattached eggs remaining in her oviduct. Whether she will live long enough to attach them is unclear. The slow failure of her chromatophore system has reached the point where the few remaining responsive patches are firing erratically, producing small flickers of color across her mantle in patterns that do not correspond to anything in the surrounding water. The lights are going out unevenly. The current passes over her body without producing any of the postural adjustments that, a month ago, would have been automatic. Somewhere above her, on the surface, the autumn light is fading from the day. The shadow under the overhang has darkened. Most of the small predators that hunt cuttlefish eggs have moved further offshore for the night. The cove is quiet. She moves slightly. One of her arms unfolds from her body and reaches toward the rock in the gesture she has been performing for 4 days. Whatever she is doing now is not visible from any angle a human observer could occupy. The arm contacts the rock, finds a corner, settles. Another egg passes through the ink-darkening chamber and emerges, attached. She rests. She has perhaps a few more eggs in her. She has perhaps an hour or 2 of effective function left. In the morning, when the light returns to this cove, she will not be moving. The eggs above her, dark on dark, will continue to develop without her. The current will continue to flow. The cove will go on. This is the cuttlefish. Sleep well.